Good morning, everybody. Welcome to our second day of conference. We're starting off with an exciting talk on genetics. I get to introduce Dr. Phil Giampietro. So Phil and I have known each other for a number of years, and it really comes because way back more than 10, 12 years ago, Judy Hall was once again trying to remind us that she is not immortal and that she needs someone to take over. And she turns around and she goes, well, I have this friend, Phil, who's really excited about arthrogryposis. You should get to meet him sometime. And soon enough, Phil ends up in Philadelphia for a number of years. We got to work somewhat closely together in Philadelphia. And actually, just talking to him now, I found out that our paths have kind of crossed a little bit of a distance. He was at Long Island Jewish before I got there. He did part of his training there. He is now up at University of Illinois, where I did my residency. But Phil is now the division chief of genetics, and he is a professor of pediatrics at University of Illinois. You can read his whole bio in the booklet, which is pretty impressive. But he just recently got the high honor of getting a named professorship. He is now the, I can never say it right, is the Ashok Ray and Purnima Ray Professorship in Pediatrics, which is a very high honor at any academic institution. But Phil is going to talk to us today about the genetics of arthrogryposis, so please give him a warm welcome.
Can everybody hear me? Well, good morning. It's very exciting to be at this conference. I've been doing it for a number of years, and I appreciate the kind invitation to be here. I hope this talk will be exciting, and I could share my enthusiasm for the field of genetics. So I'm going to talk about what's done at a genetic evaluation and how this could benefit my child. So a lot of our referrals do come from orthopedic surgeons. I've worked with orthopedic surgeons my whole career, starting with the first orthopedic surgeon that I worked with. Her name is Kathy Raggio, who I still work with today, and she's very devoted to skeletal dysplasias, and I did a lot of my formative training through the Hospital for Special Surgery when I was at Cornell. So genetics is really the science of variation, how we're all different from one each other, and here we have multiple different faces which make up our human population. Individuals vary from one another by the genes that they possess, and the physical characteristics they do possess are characteristics they acquire as a result of interactions between their genes and environment. So we use the term phenotype, which is just the outward appearance or conglomeration of characteristics that define an individual. Many people may be parents, many people are affected themselves, but some people in the audience may have seen genetics, and what are the reasons for genetic evaluation? Well, there can be many, including obtaining more precise diagnosis, offering genetic testing, enabling a better idea of the natural history, treatment options, risks for future pregnancies for parents and other family members, and help for families to work through their own ideas about causes for their child's condition. As I was talking with Dr. Mbossi, there often are parental and also provider misconceptions as well as to what a child or what an individual may have, and this is important information. Identifying a genetic cause could also help to alleviate parental guilt as well. So many people know that arthrogryposis, multiplex congenital, really is a broad array of possible causes. And this is a branching tree, but it describes intrinsic and extrinsic causes. And when we look at causes that are more intrinsic, these can include genetic factors, chromosome abnormalities, and also genes that affect connective tissue, the skeleton, the bone, and there's a whole diverse group of patients and causes for arthrogryposis that we try to compartmentalize when we see a patient with arthrogryposis. And this kind of contrast to the extrinsic or non-genetic types of factors, which can really be more environmentally based, the intrauterine environment, including uterine abnormalities, multiple gestations as well. So the causes for arthrogryposis include brain, central nervous system types of problems, neuromuscular conditions, various types of syndromic diagnoses, which are indicated in this box here, which we'll talk a little about, and I'm not going to really read them off one by one. What is a clinical genetics evaluation? Well, again, many people who have been in this audience know, but for those who are contemplating it or thinking about this for their children, these can include meeting with a genetic counselor either before the visit or certainly during the visit, and a clinical geneticist. Basically, I consider myself more of a general practitioner with an interest in genetics. So we approach our patients the way I think any primary care provider should, with detailed histories, and that includes pregnancy, birth history, developmental history, review of systems, detailed family history, pedigree taken by a genetic counselor, and then we will also look at other labs, imaging studies, MRI, et cetera, and then discuss genetic testing. Sometimes people come to the visit with genetic testing results, and they're pretty well defined, but they haven't yet sat down and had someone explain this, maybe because they moved from one place to another, but none of this has been explained. And then we work with genetic counselors to communicate the information that was derived from genetic testing, et cetera, and also discuss various psychosocial issues, how patients acclimate to the diagnosis that they have, et cetera. So what is our history? So again, history should be pretty much standard for taking care of pediatric children. I'm trained as a pediatrician, so that's my sort of my training bias. But we go into prenatal factors, maternal illness, as Judy Hall has mentioned in her papers, is very important, especially for amyoplasia, fever, maternal fever during pregnancy, as well, medications, alcohol, bleeding. What ultrasound studies have been done, and how did they show fetal movement, and were there any skeletal abnormalities? What type of genetic testing was performed? Nowadays, we are doing sophisticated testing through amniocentesis, including exome analysis as well. But there's also non-invasive prenatal genetic testing, microarray. We ask about factors like weight gain, too much, too little fluid, poly or oligohydramnios. Was the delivery natural or by C-section? And then birth parameters, if we can get them, depending upon patients and where they came from and what was done. We continue with the history, hospitalization, surgical history, developmental history, a thorough review of systems, and then family history, which includes parents' height, miscarriages, stillbirths, birth defects, other family members that may be similarly affected. And then we use the term consanguinity, meaning common ancestry. For many recessive conditions, especially the rarer ones, consanguinity is important because we then can be at risk for inheriting a common gene through a common ancestor that both parents may have. But for amyoplasia, an absence of family history is also key, because we know from studies that amyoplasia does not appear to have genetic underpinnings. So physical exam is, I see many sophisticated orthopedic surgeons in the room, but so I'm sure they pick up on all the features as well. But These include facial measurements, a look at facial structures, looking at the chin for micrognathia, as can be seen in the lower right here, and then the degree of contractures. So we pay a lot of attention to craniofacial. We look at limbs, creases. Palmar creases are very important. So babies, as Judith Hall taught me, arthrogryposis is really all about motion, really. So you think of motion and kids move in utero, right? But it's because of arthrogryposis, they're really not moving. And the creases are generated because our hands move and flex during when in utero. And when that is limited, then there are decreased creases on the palm as well. And symmetric rigid contractures are associated with amyoplasia, looking for dimples over affected joints, foot deformities, nevus flammus on the mid-forehead, radiographic information such as gracile long bones, which are osteoporotic. So lab testing, I worked with Dr. Dieterich, who is an expert in arthrogryposis and others, and we did a survey of lab testing, and really, we found that CK seems to be the more commonly associated lab finding with when you get really high levels. Those can be associated with muscular dystrophies or anterior horn cell disease, and intermediate CK levels can be associated with other forms of arthrogryposis. So again, CK, a muscle enzyme, is not a specific marker, but can be providing a clue in some instances. What other studies? I know many people may have had other studies like EMG and nerve conduction velocity. These are pretty invasive studies, and trying to do these on a child who really has very gracile bones and really have very decreased muscle mass are really very hard to do. But if one is suspecting a neurogenic form of arthrogryposis, there can be a good yield, but for more myopathic forms, the yield can be less. And people really think of it more as a second-line investigation, really. Again, this is a survey. These were part of a survey of the literature that I had worked on with Dr. Dieterich and others. But muscle biopsies now, in today's more genetic age, unless there's a clinical suspicion of an underlying congenital myopathy, muscle biopsies seem to really have little impact on diagnosis in the distal arthrogryposes. Spinal muscle atrophy and Prader-Willi syndrome, those conditions are rarely seen in association with arthrogryposis multiplex congenita. So I'll talk somewhat about genetic testing, and I know we have diverse backgrounds here, and some are really hearing this lecture and for the first time. So I may, people just may excuse me if some of this is a little basic, just to get people up to speed. But when we talk about genetics, we're talking about our genetic material, which is organized on chromosomes. Human beings have 23 pairs or 46 chromosomes in every cell of our body, and chromosomes contain our genetic units in the form of genes, and this is illustrated in this diagram here. So I like to think of use the analogy that chromosomes are more like our set of encyclopedias here, and here we have the Encyclopedia Britannica. I don't think this come kind of from the days that people knocked on your door and they sold you encyclopedias. That's where the time era that I grew up in. Now everything is electronic, so people might not see it this way, but you can think of chromosomes as this whole set of encyclopedias, and then the genes are more like the sentences on the page in the set of encyclopedias. So genes provide the instructions, and they code for proteins. And genes are then made up of DNA, so we can say that the DNA is more like the letters that make up the sentences here, and this shows the sequence analogy of the sequence profile here. So we go from higher order to really lower order. And I really started my career in genetics in the late 70s, going to graduate school. So I think I've seen pretty much the entire gamut of technology revolution over the 45 years or so that I've been in genetics. And back then, it was standard karyotyping, which then evolved into really stretching the chromosomes out to really look for tiny changes in the chromosomes. And then that was heralded by looking at just single regions through what's called FISH technology, amplifying a particular region and then getting a region to light up or not light up if it's absent or present in too much amount. And then using what's called microarray technology, which I'll talk about, but that's looking at small pieces underneath below the level of the microscope to see if there's extra or missing material. And now we're at the single nucleotide level here. So we went from 10 to the 9th billion base pairs to now the single nucleotide really in 50 years or so, which is really amazing. So how can we use this testing to help you and help provide diagnosis, which is really what I think it's all about, really. So we use the term chromosome microarray, and I don't know how many parents here are familiar with that, who may have had one of their children go to genetics and have had a microarray test. A microarray in today's era is more analogous to a chromosome analysis, but we look to see if there's variation in segments of a particular chromosome, too much or too little. We use the term copy number variation. And the indications for doing a chromosome test are many in the field of genetics, so I see other types of patients too besides arthrogryposis, but we'll use this on a child who may have developmental delays or may have multiple birth defects or facial features that are distinctive or dysmorphic or short stature, failing to thrive, or other indications. So when we do a microarray, we are just taking our own DNA, a patient DNA, and basically we are getting that, we're putting it on a plate, and we are comparing different regions. So we chop up the DNA into pieces, and then these pieces are then segregated onto different wells, and we look for each particular well, that's each segment of our genome, to see, compared to what the control DNA, this is just someone whose DNA is used as a control for the lab, is there, for each particular well, is there an equivalent amount? So here we see 2 orange lollipops, that's the control, and 2 green lollipops, so you have equal amounts for that particular region. If you're missing a region of that of the genome, then you will see for that particular region less compared to the control. So here we have 3 lollipops and 1 green and 3 orange. So you're missing a segment of that region. And then if there's a dosage gain, if there is a dosage gain, you're seeing 3 green lollipops for 1 orange lollipop. So this is just a concept, really. I think the key is to note, we're looking at copy number variation, too much, too little. And this all gets looked at in a machine, and the machine will scan. This is an example with chromosome number 7. This is the whole chromosome. And this is just showing that it's a steady state, so there's you're at the right amount of genetic material compared to a control. This is a 1-to-1 ratio. of control in patient DNA. But when it dips below for a whole region here, see this dip here? So this is below what it should be. And here you've lost a whole chunk of chromosome 7, and that's referred to as a deletion when you lose material. So this is showing us segments that are abnormal in copy number variation. And depending upon what that region and what is missing, the lab will say it's significant or not significant. So this segment has multiple genes, and the lab will look up the genes and say, oh, these genes can be involved with arthrogryposis or muscle or bone or connective tissue. And yes, we know that this region has been found, it's been reported in the literature. So they'll give us a report and they'll say that it's a significant change, really. And this is another schematic diagram showing gain or copy number gain or loss or copy number loss for deletions or duplications. And these, depending upon the region, depending upon what's known, will be signed out by the lab as significant or not significant, really. And again, another diagram here. This is a standard karyotype we call an array, an ordered array of chromosomes. And here are our 23 pairs, and we start with chromosome number 1, and we go to 22, and we have 2 X chromosomes here with the genetic sex of XX, female genetic sex. So, and this is what each chromosome looks like on a microarray as well. And this person has the normal amount of material. We don't need to go through it all, but it's pretty normal. And it's balanced, meaning that there's no extra or no missing material. So balanced is another key word that we use too. So we now can get to the level of genes. And when we look at a gene, again, this is just basic information, just to keep everyone up to speed, but we look, genes are made up of coding or exons and non-coding regions or intronic regions. So genes are made in pieces, and we start here. This is the promoter. It's a signal to make more of messenger of that particular gene so that it can be then made into a protein. So, and the DNA code is 4 letters, so C, T, A, and G, and these genes in triplets code for a particular amino acid, which are the building blocks of protein. When there are errors in the DNA, that can, in some instances, cause alterations in the protein, which then can be correlated with a particular condition. And in older times, we use this Sanger sequencing, which has now evolved into massive parallel sequencing, for which we can do many genes all at once. So Sanger is more a letter by letter by letter, and the Human Genome Project was sequenced by this. It took quite a long time, but it was done under budget and ahead of time. It started I believe it started in the 80s, and it was done in the early 2000s. So, and this is what was used. It was a really brute force sequence of our genome. So when we do the sequencing, as you can imagine, we get really gamache of the letters, the DNA, and this is just showing you this is a whole bunch of gibberish, really. And how do we interpret that? Well, it's important to then look at what the frame is, so we can then make sense here, because this is showing the frame in groups of 3 letters. The quick brown fox jumped over the lazy dog. The dog lay quietly dreaming of dinner. So now, it's only when you put this in, when you find the reading frame, can you then say, what is this supposed to be? And this is what the lab has to do. It sequences the gene and then puts it in the right frame and then tells us what's missing or not missing, really. So it's a frame of reference, really. And Many people may be familiar with the term exome analysis, when this is really more state-of-the-art in clinics now. So when we do an exome, we are looking at only 1% of our genome. It's looking at all of the genome that actually codes for protein. So if you look at the slice of the apple, the exome is really 1% of that. So we know the coding part, what actually makes proteins, is only 1%. The 99% is all of this intronic stuff that we just really don't know what it does. It has some regulatory types of aspects too. But an exome will just take out the intronic information, and then it will look at what's coding. And just to make a long story short in this slide, I'm not going to read through it word by word, but we look at variation. Now, because someone has a variation, does that mean you may get a report, VUSs, variation of unknown significance? That does not mean that that person has a mutation. A mutation has consequences. A mutation changes the protein in a harmful way to that protein. Variance can be significant or non-significant. If the variance is significant, we call that a mutation. If we don't find anything, we just say the results did not show anything. And oftentimes we get negative results on an exome. We may not have a genetic reason, or there may be other genetic causes that the exome can't identify. But uncertain results can be classified as benign or can be reclassified as harmful. So we will tell the family to come back in a year. We can ask the lab to reanalyze their exome if we don't get results, and then the lab will tell us if there's anything that's been changed or not. So this is really the type of information that we get. Now, there are probably more than 480 genes for AMC, but there are multiple pathways. These genes are part of big networks. And Judith Mulligan, who really taught me a lot about arthrogryposis, really, her dream was to really identify pathways for arthrogryposis, which can then be used as basis of treatment. So if you know the gene, if you know where it fits in a particular pathway, then you can better identify treatment modalities to help patients with that. So that's really why we study the genetics also. And this is one example of a really complicated pedigree. It must have taken, years to get this all together, really. But this was a large consanguineous. There were double arrows here, as you can see over here, which means consanguinity. Parents are closely related. And all these people here are affected. And the laboratory did a whole exome on family members in this group. And they did analysis of it, and this was the phenotype, really showing the joint contractures, and DNA analysis was subsequently done on next generation sequencing platform. So again, these are very robust. They sequence many, many genes all at once as well. It's really amazing, the technology in terms of how it's evolved. Creating genomic DNA, and then all of our genome that's fragmented, and then we then get rid of the non-coding stuff if we're doing a whole exome, and then we can then sequence all the short segments of DNA and look to see if there are any known variants. And this is just an algorithm, but the process of doing whole exome really is, I'm more the clinical end of things, but we interface with the lab. On one side, the lab does the analysis. They don't see the patient. We see the patient, but there's a referring doctor on the other end, or a referring provider that we have to, get back to, and we also have to get back to the family as well. So it's a whole complex array of, interactions as well. And, we kind of, exome reports can be quite complicated. I don't know if people in the audience have gotten them, and if parents or family members or providers here look at them. And, it can take me several hours to look at a report if we get multiple variants and looking up the papers and looking to see what, the genes do. And basically, I consider myself a matchmaker or a phenotyper. So I'm looking at a report that I get from the lab, and then it gives me a set of findings associated with a particular gene. And then I have to go back and I have to read my report on the patient and what other people found. And I just say, are the two, together, or are they far apart? Is this something that I can't put together? And that. So that's basically how I spend my days, that's kind of all I do, really. And so this gene that was identified in this family codes for a cycling membrane protein, and it has to do with protein synthesis as well. But this is just an illustrative example of whole exome, which is used for a variety of diagnoses, ranging from developmental to dysmorphic to birth defects. And the yield is really approaching, this is an older kind of slide, but 7 years older. I think it still depends on the condition, but we can get yields of up to 50% depending upon the cause. For autism, it's about 40 to 50% doing exomes as well. And now there are panels, and I'm just going to kind of say it's been my experience. I don't use panels really anymore for arthrogryposis, and some people out there, may have had their child tested with the panel. I think now in the clinical setting, I know, again, Judith Hall has trained me in this area, says go for a genome, and I think that that's true, that's the ideal. I know in the United States, we can't get genomes most of the time in clinic. We rely on exomes. Many insurances will cover an exome, at least where I practice. We had one lab that was doing them for a while, but it really became hard for us to get our patients into that. So most of the time we're doing whole genome, whole exome analysis as well. And this is just an example of patients that have been solved. Their mysteries have been solved through the use of whole exome and next generation sequencing. This one patient here has a deletion or missing 22 nucleotides in a particular gene, the STAC3, and this patient here has a mutation in filament C. Okay. So we've thrown the terms whole exome and whole genome. Whole genome means you sequence everything in the kitchen sink, really, the whole kit and caboodle. You sequence the introns, the exons. So in terms of variation, if you just look here, the bioinformatics, this is not done by someone sitting at a desk and paging through. This is done by bioinformatic analysis. So the computer is actually manually sorting out all different variants, and they go into like bins. This is commonly seen in the population. This is rare in the population. This change correlates with the patient's features. This doesn't. So this is all being done by machine. And I trained, in older times where you just sat and you looked at books and you would get results and you would be able to, you would just be looking at limited numbers and you did all this. But just imagine, you're getting tons of variants that will take you more than your lifetime to just do one person, really. So that's why we rely on sophisticated computer bioinformatics. And I'm saying most of my experience is a lot of insurances now will cover exomes, but still not all. And exome sequencing is really good for more of the single gene types of conditions, whereas whole genome is more for the more complicated conditions. And this one paper showed 33 reported studies using whole exome and whole genome with the yield approaching 60%, and most of the variants are in the coding regions of the gene. So this slide shows the different types of testing, and we go from more narrow in terms of focusing by panel to more genome-wide interrogation, really. And if we Again, I think we're kind of here now, at least in my own practice, I'm kind of here. I don't use panels, really. I just go with an exome, and I go with a more broader test, really, because there are so many overlapping clinical features in all the arthrogrypotic features. It's really hard to really pick them out, really. There's so much overlap, really, and, you know, so, but when you go here in terms of genome-wide and sequencing everything, then, you know, the phenotypic capabilities then become, you know, less, more non-specific. You know, so you trade maybe sensitivities for specificity, but I think in the end, it's better for the patient and that. And the good thing is many of these tests now can be done. You don't need to poke people. You can just do it with a cheek swab, really, and get the cells that you need. So this is also encouraging. I just wanted to give hope to people and say that we haven't learned all there is to know about the genetics of arthrogryposis. This is a 2022 paper, and we don't need to go through individual genes, but this basically shows about you know, all these genes are new genes that have been identified, really, in this one person's series of cases over here of 315 patients. So we don't know all there is to know about the genetics, and we're still learning more. Okay, so, and in their series, most of the time, these genes affected the skeleton, as you can see here. Some affected the neuromuscular junction, axial glial cells, and by and large, the inheritance was autosomal recessive, followed by autosomal dominant and X-linked genes on the X chromosome. You can get more complicated pictures. I don't want to go into the slide too much, but we talk about identifying the gene for a particular person's condition. But it can actually be several genes, meaning oligo, meaning few, and there can be more complicated scenarios where you have two genes or so that may be important, or modifiers. So why is someone in one family, in the same family, a different family member, why are they having different features? And those can be due to environmental or other types of modifier effects. So, you know, we try to solve the puzzle, and we then go through a process of genetic counseling. We'll talk about how this happened, how was it inherited, what about prenatal diagnosis. Does the family have the psychosocial resources to really, you know, move and live with their condition as well and live with the genetics of this? Have we helped reduce the family's anxiety in some way? We synthesize all of this, and we try to provide more precise diagnoses for families, and again, realizing that not all forms of arthrogryposis are genetic, some such as amyoplasia are not. And we provide all this information to the family and providers. Insurance issues can still be problematic, and I don't know, I feel that it's gotten better in some ways for genetic testing, but there are still some insurances that feel a lot of this is experimental too. So our genetic counselors and myself have to kind of do peer-to-peer types of insurance. Genetics, you know, is not as simple as I thought it would be when I went into the field, and it's not getting any easier, actually. It's getting fun, but it's not getting easier. And I just hope that this talk may have provided some optimism that we're still learning more about arthrogryposis as well, which can help hopefully translate into better clinical care for families and providers. So thank you. And,